right, everyone. So I'm happy to say school is almost over. It's been a great week. We've really enjoyed having you here as our guests. We've learned a lot from you. I hope you've learned a lot from us. So for our last lecture, I'm really proud to be able to announce Professor John Yates as our speaker. So I just want to say a few words about him before we bring him up. You know, when I think about the field of proteomics and mass spectrometry, he is one of the people who really built this field. And he's made so many contributions. And if you think about all the things we learned this week, he's had his hands in just about all of them. And you know, he's got a set of papers that have just changed how we do things. And you know, one of them that I think you should all look up and check out is a paper written in 1986 in PNAS when he was a graduate student. You know this one? Yeah. If you read that paper, it describes everything we do today, and it was written in 1986. And it's just incredible. It was a decade ahead of the field, at least. And it just laid out exactly what we talked about all week. It was way ahead of everything else. It was just incredible. So you should read that one and cite it, because I don't think it gets cited as much as it should. It gets a lot of citations, don't get me wrong. So after he left graduate school with Don Hunt, he went on to do a postdoc with Lee Hood, and he started his career at University of Washington in Seattle, where he made another remarkable contribution there with the sequest algorithm, really the idea of how we take tandem mass spectra and how we automatically match them to sequence. And before that time was pretty much done by hand, and it really changed how we can do things, right? And it was revolutionary. And then he moved to Scripps, and there's so many other things. I would just point to another landmark paper that changed how we think about proteomics is the mud pick technology and how to do two-dimensional separations and to couple that online. That was also transformative. There's so many other things, and I won't embarrass him with all those things. I just know that Professor Yates has really invented this space that we're working in, and it's so great to be able to have him here to cap off this week. So he's won many awards for that, and he's also right now the editor of the Journal of Proteome Research, associate editor at Analytical Chemistry, I think, and was. Okay, you stepped down to be the lead editor at JPR. So send all your stuff to JPR. Professor Yates.
Can you hear me? Is it on? Can you hear me in the back? Okay. All right, Josh made the mistake of not telling me what to talk about. So I figured end of the week, you know, you've been hearing about p-values. Is it 0.05? Is it 0.1? Is it how many n values are we supposed to use? So you've been hearing about all that all week. So I thought maybe I'd make this talk a little bit lighter than what you've been hearing this week and talk a little bit about sort of the history of proteomics, kind of where it came from and what my role had been in doing some of these things. And I'll tell you maybe some of the lessons I've learned along the way, various things. This is a great quote. I love this quote because it really defines how tools help really drive science. And it's not so much about the concepts, it's more about having the tools to be able to discover new things. I love to do discovery. I like to learn new things. But sometimes the development of a new tool comes about because you have to, because you are at an end. Something has happened that you need to have a new tool to do something. And this is what happened to me at the University of Washington when I was there in the 1990s. This is security footage outside my lab, which caught this guy stealing my autosampler. I don't know if you recognize him, but the lab down here is that's the Abersold lab. So with our autosampler gone, we had to think about new ways to analyze proteins. and you know, the grad students and postdocs are not going to sit there and load single proteins into columns and then run them. So we had to think about how to do new things. Okay, so I love this article, which came out in, look at the date, 1978, the decline and fall of protein chemistry. And so what happened in 1977? We're going to do this kind of like a Trump rally where it's going to be call and response. So, of course, 1977, Sanger publishes paper on sequencing DNA. Maximum Gilbert publishes paper sequencing DNA. And this was somebody saying, well, protein sequencing is dead. So, interesting story. I got my PhD from Don Hunt. Josh did his postdoc with Don Hunt. And in early 1977, Don Hunt goes to Cambridge University to do a sabbatical. And he's in Dudley Williams's lab. And Dudley is a famous organic chemist who also does mass spectrometry and stuff. And Dudley goes, so, Don, what do you want to work on for the next year? And Don says, well, I think I'm going to develop a mass spec-based method for sequencing DNA. Dudley nods his head and says, hey, you know, they have this seminar series over at the Medical Research Institute, Medical Research College, MRC, over in Cambridge. And there's a lecture coming up you really should go see. And so Don gets on his bicycle and he rides over to the MRC and gets in this big auditorium and it's packed. And there's Fred Sanger giving a talk, sequencing lambda phage or whatever it was. And Don goes, oh no, I guess I gotta do something different. And so Don decides he's gonna work on sequencing proteins by mass spectrometry. But I guess he hadn't seen this article. So he didn't know that that would be a waste of time too. Okay, and so 1988, this report comes out. We've got to sequence the human genome. 1989, human genome projects are looming with the future. Protein chemistry was uncertain. I went off to do my postdoc with Lee Hood, and Lee Hood's lab, I don't know if you the history of it, but he was developing the 4-color DNA sequencer, which was really prominent in the early days of the Human Genome Project. And everything in the lab, Lee had a big lab, he had 70 people in that lab. Everything in the lab was DNA sequencing this, DNA sequencing that we're gonna do. How are we gonna do DNA sequencing? We do shotgun sequencing, we're gonna do directed sequencing, so on and so forth. And there were a huge number of very prominent people coming through the lab. Watson, guy from Washington University, the guy from the Sanger Center, you know, all these prominent people in the field of DNA sequencing. And so I'm sitting around thinking, what am I going to do with protein chemistry? And Lee still had a small group within his major operation that was doing sequencing. And if you read the report that came out of the National Research Council, and the stuff in red is probably the important part. So as visualized, it would also lead to the development of a wide range of new DNA technologies and produce the maps and sequences of genomes. So what's missing? What are they failing to tell us about? They're failing to tell us that this is going to be important for protein biochemistry. So I'm thinking, I'm at Caltech, and I'm thinking, what would the role of protein biochemistry and hence mass spectrometry play in the area of genomics? And I went to Caltech. So this was, Caltech undergraduates are absolutely brilliant. I know University of Wisconsin has great students, but Caltech students are fantastic. So in the 100-year anniversary of the start of Caltech, they did this to the Hollywood sign. So at the time, the major way to sequence proteins was the protein sequenator. And this was developed at Caltech. It was developed by Lee Hood and by Bill Dreyer. Bill Dreyer famously does not get any credit for having invented gas phase sequencing, but he invented it. Lee made it work with Mike Hunkapiller, and it became known as the protein sequenator. The ABI was founded around this technology. They sold thousands of these things. They were great instruments. And one of the things that Rudy Arbusel did when he was a postdoc at Lee's lab is he started sequencing spots off of 2D gels by the protein sequenator. One of the things that I wanted to do when I went there was to take spots off 2D gels and analyze them by mass spectrometry. Okay, so the fight on the mass spectrometry end had always been with the Edmunds sequenator. And now we got this problem with genomics coming along. And this is 1989, and 1989 was a 88, 89 was a pretty big year for protein chemistry, for mass spectrometry. What happened? Electrospray ionization was invented, 87, started to appear in labs in 88. Laser desorption came about, and so protein chemists gained a new analytical tool. This is Research News and Science. They had this nice little box, mass specs move in on protein sequencers, talking about it. This is kind of a depiction of a particular kind of mass spectrometer, tandem mass spectrometer. I'm highlighting some important opinions here. In Hunt's vision, the future of the protein chemistry labs will have 2 mass spectrometers for every sequenator, where now you're lucky if you find a mass spectrometer for every 6 or 7. But his critics argue that no one but Hunt has been able to extract sequence data from a quadrupole. So even within the mass spectrometry field, we're having these big fights. And I love to put up this quote. So others are waiting for electrospray or laser desorption to be adapted to magnetic sector machines, which have greater resolution and a more proven record in protein sequencing. When that happens, the sky is the limit, predicts Steve Carr, a mass spectrometrist with SmithKline French Laboratories. So the force-sector mass spectrometer, magnetic sector mass spectrometer, was the Orbi-Trap of its day. What do they look like? They look like this. So they fill up a room, they're big, these are 1.5 ton, 2 ton magnets, and they cost $1.5 million. So if you were important in mass spectrometry, you had one of these babies. And what happened to them? Well, electrospray ionization came along, and just like the dinosaurs, they became extinct. So they did not adapt well to the ESI source, because the source the ion source on a magnetic sector instrument is floated at 10,000 volts. And then they also scanned very slowly. They would do one scan in about 3 minutes, 4 minutes, and so it was too slow for LC, unless you do really bad LC. Okay, so mass spectrometry, I knew this, and others in the field, we all kind of knew this, that it's unbiased and potentially very fast. We could increase the scale of measurements, increase sensitivity of measurements, generate new types of data, so it's good for PTMs and things like that, and increase throughput. More experiments is a function of time. But in the field of mass spectrometry, data analysis was the bottleneck. And in fact, the data analysis is such a hard problem that it created kind of like full employment for mass spectrometrists. So there really was not a lot of incentive to make it easier, because it got us all jobs. In fact, Don Hunt started at the University of Virginia in 1968 because they had to hire a mass spectrometrist in order to get a mass spectrometer. Okay, but there has always been a lot of interest in using computers in mass spectrometry. So Beeman's lab, Klaus Beeman, the now deceased, but he was very prominent in both training huge numbers of mass spectrometers, but also in developing all the things that we kind of do today. And so he was a leader in this area. He was at MIT, and MIT was a leader in the development of computers. And so it only made sense for him to also start to adapt that technology to mass spectrometry. And so he developed methods to acquire, display, and store MS data. So total ion currents, selected ion currents. These were, revolutionary things in their day. And he also started to develop data analysis methods, including library searching and later on peptide sequencing algorithms. McLafferty's lab was also very involved in developing library searching. In fact, there was a battle going on between the two, and McLafferty was extremely aggressive at going out and if you acquired a spectrum for some molecule that you'd interpreted, he would try to get the spectrum from you, and so he could add it to his library. The other interesting thing at this time was Joshua Lederberg. And so Joshua Lederberg got a Nobel Prize in medicine, I guess, for microbiology, showing that bacteria have sex. And he was very young, so he had a whole career ahead of him to do lots of different things. And he was at Stanford, and he got interested in AI. And he thought that mass spectral data interpretation would be a great intellectual problem to develop AI algorithms. So they started working on this algorithm called Dendral back in the day, and it actually made a lot of progress, but the main challenge for them was that computers just weren't very fast. And one of the things that Beeman's lab had was they had this IBM 1800 instrument, which fills up a room. They had to have a lot of space back then for their mass spectrometers and for their computers, but that thing probably wasn't as powerful as the Apple Watch I have. All right, so people also were trying to develop ways to interpret peptides. So de novo analysis was largely limited to a spectrum and was not particularly accurate or general. Data quality was always the main issue. These are some of the papers that came out this time frame, 1988 or so, where people were trying to sequence peptides by computer interpretation. But by and large, the way that peptides were interpreted was by this. And I used to go over to the computer center this is my interaction with computers, go over to the computer center at UVA, and I would get reams of computer paper, and I would use that as scratch paper while I was trying to interpret tandem mass spectra. So I always had a big interest in trying to develop computer algorithms to help with the interpretation of the data. Note the computer right here. My wife kindly allowed me to go buy a PC for $1,800, which was this thing, and I used it to try to write computer algorithms to help with data analysis. So one of the things we eventually did published was an algorithm together with Finnegan Matt on interpreting data. But one of the things I got interested in while I was still at Virginia was a program called PC Gene. And this was developed by the Swiss Prot guys, and they converted it to a PC. And we bought this, and it came from a company called IntelliGenetics. And they had the contract for GenBank. And so at this particular time, if you wanted to connect to GenBank, you had to call into a Telenet node on that number, and then that's how you got access to sequence data. But I got this program, and I started using all the programs that were within this thing to try to characterize, try to help with the analysis of proteins. And one of the things it had was a database. And what I learned was that I could use that database as a way to help me interpret mass spectrometry data for proteins. And this is where I started getting interested in how one could interface databases with the interpretation of mass spectrometry data. And so I'd written this little applications note for IntelliGenetics. They were having a contest on interesting uses of their tools, and they were going to have us all fly out to San Diego for some meeting, and they would have a dinner and all this stuff, and they decided that was too expensive. They just sent me a check for $300. Okay, and that was fine too. I could use $300. Okay, so my aha moment in terms of this. I've been thinking about that, and when I finally went off and started my own faculty position, we were manually sequencing class 2 MHC peptides in the lab. We would partially sequence 4 to 6 residues, then we would blast search the partial sequence by emailing the sequences to the blast server. And that was found in NCBI, and we would use the hits to make sure we weren't spending time on known sequences, because there were the whole families, and they were all very related and things like that. So we wanted to make sure we weren't just sequencing something that was already a known sequence. And so while sitting there waiting for a blast search to come back, I wondered, why not just search the sequence database with Spectra directly? So why not just take the MS Spectra and do that? Okay, so that's a fantastic idea, right? So the problems are, how do we get data access? So we had to access the MS data, we had to access sequence data, and just for perspective, how many people know what Netscape is? Probably if you're over a certain age, you probably know. Okay, so Netscape, actually, this was kind of what started the World Wide Web, and its launches 1994. I'm starting my faculty position 1992. So we're still using FTP as a way to get access to things like sequence data. And then how do we compare the spectra? So there have been methods to compare spectra for libraries searching, but this would require comparing sequence to spectra, and then how to reconstruct that spectrum from the sequence so we can do some kind of comparison. So there are lots of problems. So all the mass spectrometry data, which you probably know if you've tried to do this now, is it's proprietary, and so it's in a encrypted format. And that's not because the mass spectrometry manufacturers are evil people. It's because the government requires them to do that. You're not supposed to be able to access the raw electronic data because then you could actually go in and change it. And the FDA requires that you not be able to do that if you're going to submit that data for a drug application or something like that. And a programmer in my lab demonstrated to me that he can get into a manufacturer's file format and change the data. So it's not as encrypted as they think it is. Okay, so the data access problem. So we had limited access to data in 1992. There was this thing called NetCDF. And it looked to me like it was a manufacturer's slow walk to appease noisy people in the field that wanted access to the raw data. You could only output a single spectrum. And Finnegan had given me a program that allowed me to output an MSMS to a flat file format, but it was very difficult to do an entire LC MSMS run. But at the time, DDA didn't exist anyway. But you could still do 10, 12 MSMS in a single LC run by typing things in and being really fast. And you'd have to use scan numbers as a way to bracket the file to put things together. It was very, very cumbersome. And I was having a lot of trouble getting the attention of Finnegan. Finnegan is the former Thermo. So Finnegan Matt became Thermo Electron, became Thermo Instruments, became Thermo Fisher. So the way we got their attention was that Jimmy Yang, who was a programmer in my lab, he cracked the Finnegan DAT binary file format. And once I told them that, they immediately sent me an NDA and then sent me a program that we could use in order to get access to the data. All right, so the genome projects were launched probably about 1990. Protein sequence data was coming. Eventually they sequenced all these things and then some. So we could, by 1993, we could download databases like GenPep by using FTP. And remember that Haemophilus influenza genome, this is the first genome sequence, was published in 1995 by Ventnor's Tiger Institute. So we could get access to the databases, and anybody know what FTP is? File Transfer Protocol. Yeah, so the world's changed. Okay, so this is how we're going to compare sequence to spectra. So step one, we have to pre-process all the tandem mass spectra. And we have to, the data back then, this was triple quad data, so it's a lot noisier than probably what you're used to now. So we do this. Step two is we search the database, we calculate molecular weight of peptides in the database. We have no reference to proteolytic cleavage, and primarily because we're still working on MHC class 1, class 2 peptides, and so they're not actually created by any known proteases. We do a preliminary scoring, ranking of scores. This is a formula we came up to try to compare spectra. And then we do a spectral reconstruction, and then we came up with this idea of doing a cross-correlation analysis. So we do noise reduction on the MSMS data by removing all but the 200 most abundant ions. We divide into 10 equal sections, and ion abundances are then normalized. So it winds up looking like this. See, it's very noisy. This is triple quad data. It's very noisy data back then. And then we reconstruct the tandem mass spectrum from the sequence by trying to replicate that. And we give everything an abundance value of about 50%. We try to put in the neutral loss, water and ammonia in here. And we give B and Y ions, so where they come from the N terminus or the C terminus, we try to give them the same values associated with it. So we create this thing. And this was done in part because we could not predict the abundances of fragment ions based on sequence at the time. And people have tried to do that since then with some varying degrees of success. And we thought we could eliminate that problem just by doing it this way. And I think it winds up working pretty well. And then we needed to have a way to do a comparison. And we stumbled upon this paper by Kevin Owens. He was at Drexel University at the time, but this is some work he did at Drexel. And he came up with this algorithm, and it was based on, which was published in Applied Spectroscopy Reviews 1992, where he's using correlation analysis techniques to mass spectral data. It really came out of Hiefja's lab, who was using cross-correlation analysis to compare infrared spectra. And Kevin was a graduate student in Hiefja's lab back in the day, and so he continued working on cross-correlation analysis as a way to compare mass spectral data. And it actually provided us with a lot of insight on how to do these comparisons. So you use a fast Fourier transform as a way to do these comparisons in an inverse FFT, and when you do that, this is what the comparison between these two spectra looks like. And then what Jimmy did is he took an average, or he took the central point, and then he subtracted off the noise around plus and minus 75 around the 0 point value as a way to come up with a cross-correlation score. And that turned out that worked pretty well. So we had shotgun sequencing in our first paper published in JASMUS 1994. We're getting great scores for various spectra, but we're not acquiring all that many spectra at the time, and the spectra that we do acquire are actually pretty good quality. So this is another paper. This is doing E. coli. The previous one was yeast. And so we're doing shotgun sequencing, but we don't really have a way to automate the acquisition of tandem mass spectra at this point. And so it's not really all that effective, except that it's just kind of interesting. So of course, what do you do when you've got a new thing, right? You go to NIH, you ask for money. And the second mouse gets the cheese.
What happens to the first one?
I'll let you think about that a little bit. All right, so I submit a grant. And this is actually the revision of the grant. And I get an awesome score, 287. This is a score of 100 to 500. And so that's percentile 74.3. So the lower the percentile, the better. So I'm getting killed here. And what the reviewer says is, you shouldn't do it this way. You should do de novo sequencing, write an algorithm, do de novo sequencing, and then use FASTA as a way to search the data through the sequence database. And one of the reviewers just happened to be the guy that developed FASTA. I'm trying to get this thing published too. Okay, so I sent it to PNAS, sent it to Fred McLafferty. I said, Fred should understand this because he knows about library searching. He'll get it. And I think he's a reviewer, and I know who the second reviewer was because he revealed himself to somebody who then told me. It was Klaus Beeman. And this is the second letter from Protein Science. So it's been rejected twice from Protein Science, rejected from PNAS, and then it eventually gets into, Mike Gross takes pity on me, and he takes it into JASMUS. And note the, note the date here. This is February 1994. All right, so it finally gets into JASMUS, and it's now the most highly cited paper in JASMUS by a factor of about 6 or 7 or something like that. So Mike made a good call. All right, so we very quickly followed that up with showing that we can search modified tandem mass spectra through databases, and in particular phosphorylation. Probably not the most efficient way to do it, but we show that we can in fact identify things which are not actually in the database using mass spectrometry data. This, of course, these things, of course, excite get phosphoproteomics, large-scale phosphoproteomics going. We also showed in this paper that we could selectively pull out phosphorylated peptides using a neutral loss scan algorithm. I'll talk about this in a minute. And then this is just a spectrum. One of the first things we ever published using the algorithm to identify it. This was done with Mike Gelb's lab at the University of Washington. You can see the peptides, the phosphorylation groups, and so forth. We also show that we can search nucleotide sequence databases. So we're doing a 6-frame translation, and we're scanning through it and matching spectra up. One of the interesting things about this is that when you convert to a 6-frame translation, the numbers go up dramatically, huge. And we were able to still match things up using the method, which gave us some confidence that the technique was pretty sensitive. And we used that in a study on Plasmodium yoeli to try to find ORFs. We did what is now called proteogenomics, so we did all stop-to-stop ORFs were created and then searched to help identify the genes. And it became actually quite useful because this was a AT-rich genome, and so the traditional GC or finding algorithms weren't working very well. So that helped with this. We also published a paper where we looked at amino acid sequence variations in proteins, and we used that to look at hemoglobins. Doing this, we had an algorithm which we called sequest-SNP, which was looking at conversions of nucleotides within the program. But there are probably better ways to do this now. This was not a very efficient algorithm. All we wanted to do was do proof of concept by that point. Okay, so by 1996, we have the ability to identify peptides and proteins, modifications, and assist in gene annotation. We've also published in 1998 a library search method using some of the techniques that we developed. There are now general and specific libraries that are available. What we need are completed genomes. They're coming. So we also need faster scanning mass spectrometers. So the LCQ was introduced in 1995. And so the LCQ will do 3 MS-MS scans a second. What we're doing on the triple quad is we're doing 1 MS-MS scan every 3 to 4 seconds. So we have to accumulate data. We also need faster computers. And so we built a compute cluster, a Beowulf cluster. They were called Beowulf clusters back then. And this was actually kind of fun to do this. We continue to use, everybody's got clusters nowadays. Data acquisition algorithms and data transfer, so we got that done. We're starting to think about larger scale chromatographic separations, LC-LC. I've always been enamored by the concept of LC-LC. And we're also starting to work on biochemical methods suited for mass spectrometry. So we want to be able to digest and process complex protein mixtures, because really people had not thought about that at the time. How do you digest a complex protein mixture and get everything digested? Okay, the other thing that comes along, this is introduced with the TSQ 70, and it's kind of like this very dormant technology. So it's in the TSQ 70 came out in 1986 or so, and this technology is sitting dormant within this instrument. People know it's there, but they don't really know what you can do with it. And it was developed by, I think Urs Steiner is the brainchild behind this. This is the patent application that they submitted. What it creates is instrument control language. So it's this ability to write computer programs to control the operation of the mass spectrometer. And it's really not until almost half a decade later that people start trying to exploit this technology to do things. And this is a lesson that I learned is, so about 1994, 3 or so, I asked an engineer at Thermo, can I use instrument control language to write a program to automatically acquire tandem mass spectra? And he told me, no, you can't do that. That won't work. And I said, okay, he's an engineer. He's at Thermo. What do I know? And so this paper comes out showing that you can select ions out of a chromatogram and do automatic MS-MS on it. So this comes out of Terry Lee's laboratory. He's at the City of Hope at the time. Program written by Mark Davis, I guess, or Doug Stahl. And they do automated capillary liquid chromatography, mass spectrometry for peptide and protein characterization. Very noticed paper, but they're doing they're doing DDA, data-dependent data acquisition. Okay, so I of course see it, and I'm kind of mad, but I can't do exactly what they did because they just published it. And so we do this other thing where we couple together a neutral loss scan as a way to trigger an MS-MS scan to look for phosphopeptides, and we publish that. But we publish it within that paper searching modified tandem mass spectra, and so it's totally lost on anybody that we've done it. But we got out into the literature, data-dependent data acquisition, just for a different reason. Okay, so that's the paper there. It's also 1995. So the other thing that I always find fascinating about science is that you never know what's going to be useful. And Jim Jorgensen, if you guys know Jim, he's a brilliant chromatographer, one of the most creative guys in chromatography, I think, that's ever been. And they're trying to figure out wall effects in chromatography. So we'd have big columns. You get most of the molecules that are going down the center of the column separate quite nicely. the molecules that are going down the wall are not separating as well. And so there's this thing in chromatography called wall effects. And so to try to better understand wall effects, what they're doing is they're trying to decrease the size of the column so that the diffusion limit is much smaller than the size of the column or something like that. And so they're starting to build these columns with inner diameters from 20 to 50 microns. And so this paper comes out in 1989 or so. These guys have no idea about mass spectrometry. And turns out that this stuff just dovetails beautifully into what is needed for electrospray ionization. The other thing is, so this is a 1984, J. Calvin Giddings, also another brilliant separations guy. He's talking about two-dimensional separations, and he kind of lays out all the sort of things you need to think about in order to do effective two-dimensional separations. And here's Jim Jorgensen putting together a system. I know this is a little hard to read. It's putting together a system to do two-dimensional separations, and they've got all these valves in here for directing flows in various configurations, but they're using a UV detector to do these separations. And this is published in 1990. So I'm super interested in this stuff. And so I go out and I buy this thing from Perceptive Biosystems. This is designed by Fred Vernier. I think it's got 3 10-port valves or something on it. And so you can do all kinds of very fancy separations on the system. This was sold to me by Ken Miller. Any of you know the head of marketing at Thermo? What?
Is it the integral?
Yes. Yeah, this is a great system, but way beyond what I actually needed. And so it's fairly uses fairly large diameter columns. I think we're doing 1 millimeter columns or something like that. Fairly high flow rates, 100 microliters a minute or so. And we do our first major large-scale shotgun proteomics experiment. And we identify 189 yeast proteins using this system. And I know that doesn't sound like a lot, but the record at that point in time was 160 proteins, which were spots all picked off of 2D gels. And that was done by Matthias Mann. And they were using MALDI time of flight in order to identify them. And I'm sure that took them many months to get that completed. We did this in 24 hours. So clearly there's a problem because there are way more proteins in yeast than 189 proteins. And I'm thinking it's the scale of the chromatography. And so we rethink how to do the chromatography. We come up with this contraption to do it, where we take a few silica capillary column, we pack the front end with reverse phase resin, the middle with ion exchange, and then we do reverse phase at the top. This is really just a desalting phase. So we get the peptides to stick to the strong cation exchange. And then rather than doing a gradient across the strong cation exchange, we just do salt bumps of increasing amount of salt. And so we move populations of peptides from here to there, and then we separate them in the mass spectrometer. So you could do 10 or so fractions, 10 or 12 fractions onto the reverse phase and then into the mass spectrometer. So you can effectively do a two-dimensional separation, but it's not in a true sense that Jim Jorgensen would do. In fact, I'm not even sure they recognize this as two-dimensional separations. But anyway, we do our yeast proteome experiment. We call this, what we wanted to call it originally was mud pit, multidimensional protein identification technology, but one of the reviewers said that name is very adolescent and I don't think you should use it. And so we didn't. But Mike Washburn has a stiffer spine, and he said, we're going to use it. But one of the reviewers didn't complain about it anyway. So now we get about an order of magnitude more proteins using this strategy, and we're actually trying to be very conservative in how we identify proteins in this process because we don't have any of the statistical algorithms that people now use as a way to calculate things. So we actually went through all the spectra by hand. Okay, so So now this is where we are. So we have, we're able to take a protein mixture, digest it with trypsin or something, and put it in the mass spectrometer, separate it, automatically collect spectra, automatically process those spectra, filter the spectra, and reassemble the protein identification. So now we have this process for doing shotgun identification. And one of the things that this is sort of defining what we might want to do relative to genomics, because these are the things you can't get out of genomics. So post-translational events, what changes are occurring on the protein, regulation of function, how's the protein's activity regulated, covalent modification, protein localization, protein-protein interactions, protein-DNA interactions, temporal, spatial, quantitative parameters. And so now we've got the ability to play, to figure out what proteins are doing, what their functions are, because bioinformatics is not going to tell you what a protein does. And so across this time frame of 1989 to 1994, we have this convergence of technologies, sequences, mass spectrometry, ionization, computational algorithms, and out of that we get proteomics. And Stan Fields thinks that proteomics is one of the great unintended consequences of genomics. People didn't anticipate that it was going to happen, but it happened, and it's actually been a great thing that's come out of proteomics. And I would concur with that. I think it's been fantastic. Okay, so I want to tell you, I told Josh that I would tell you that stuff, and then I was going to tell you some new stuff. And so I'll quickly go through this. This is just some stuff that we've been doing recently that's kind of interesting. We just published it. It's a collaboration with Pepe Ping's lab at UCLA. So we look at doing quantitative temporal analysis of protein dynamics and cardiac remodeling. And so we've been playing around with these new technologies for bioorthogonal chemistry. A method called BondCat, and it's based on azidohomoalanine, which is a methionine analog. Looks like this. If you are up on your bioorthogonal chemistry, this is click chemistry. And so AHA can be accepted by endogenous mTRNA and inserted into newly synthesized proteins in cells. This was shown by Dave Terrell's lab and Aaron Schumann's lab back a number of years ago. So the AHA NSPs throughout the proteome can be tagged with biotin by click reaction, and you can enrich them on avidin beans. So once you have a labeled protein with AHA, you can use this click chemistry, click it to biotin, and then use the biotin to use the avidin to pull out any proteins that are newly synthesized. So one of the things that we did, our contribution to this area, is that we had synthesized a stabilized isotope labeled version of this so we could do better quantitative proteomics. There is a heavy labeled biotin you can use for this chemistry, but that occurs after the fact, and so we wanted to try to develop a molecule that we could use a little bit earlier in the process. This was synthesized by William Wood and Salim Bakalala at Cambridge Isotope Laboratories. It wound up being a huge albatross on their neck because it took them, I think, 2 years to make this. Even though there was a recipe in the patent literature, there was a contaminant they couldn't get rid of, and we paid them a lot of money to make it for us, and I think it cost them twice as much to actually make it. So I think we wound up, we ended up on the upside. Okay, so most proteomic analysis in animal models of disease are used to learn the etiology of diseases, and a lot of times what people do is that they look at the diseased end, they look at the normal end, and we oftentimes use stabilized isotope labeling as a way to compare between normal and disease. And usually this is end-stage disease, and we're often looking for a signal in a large background. And some of the proteomic changes may be increasing over time as you get closer and closer to disease. And so our hypothesis with that, we would be able to use AHA as a sensitive proteomic method for animal models of disease. It would allow us to look at various stages of disease and pull out what new things might be synthesized in relation to a particular disease that's going on. That's the theory. And so we had to develop, people had not done this in live animals, at least live mammals, and so we had to figure out how to do this. And so we just consulted with a nutritionist at Harlan Laboratories, how should we change the feed? And she just said, well, remove methionine and then add your AHA. So that was a lot of help. And actually, I think this is actual food. So the control is orange and the AHA is blue. And the animals still eat it. And so this is the process. We pulse in AHA, labeling in mammals. We call this POM because mass spectrometers like acronyms. So we label, we do tissue extraction, protein extraction, click chemistry, we digest, we enrich. And we published a paper where we showed, we looked at a couple of different ways to enrich, and what we found the best strategy is to digest the proteins after they've been clicked, and then to enrich the biotin labeled peptides. We get better recoveries off the avidin by doing it that way rather than trying to digest intact proteins off the avidin. Okay, and so these are heart failure models, and what happens is that the muscle in the heart starts to weaken and becomes thinner, and it can't push the blood out up to the aorta. It starts to pool, and it starts resulting in this cycle that ends up in heart failure. Okay, this is heart failure across the US. And notice that it correlates quite well with how you vote in elections. You guys up here in Wisconsin are not too bad. The other thing that seems to correlate quite well is income. So if you look at the coast in California, where it's the wealthy part of the state, you see it's all pretty much low heart failure rates, but you go out to some of the regions which are not as wealthy, you see much higher rates of heart failure. And if you look at, let's see, Mississippi's right here, very high rates of heart failure, Upper Texas, Wisconsin, not too bad. And I want somebody to explain to me why Wisconsin lost this part to Michigan. I know it's the hand, and that somehow you got the upper tip. There are actually a lot of interesting stories, I think, about how the state lines were drawn. So I've been trying to get somebody to explain to me why Texas doesn't own this part of Oklahoma, or why Oklahoma doesn't own this part of Texas. So one of the explanations will do. Okay, so about 5.6 million adults in the United States have heart failure. 1 in 9 deaths, 2009, included heart failure as a contributing cause. About half the people who develop heart failure die within 5 years of diagnosis. Heart failure costs the country about $30 billion. This includes healthcare, medications, missed days of work, things of that nature. All right, so what happens as this occurs is that you have the normal heart, you get adaptive remodeling, then maladaptive remodeling, and then you wind up with heart failure. These beta adrenergic receptors become overstimulated over time. It's one of the common stages before heart failure in many diseases as cardiac remodeling. And you get changes in myocardial structure and function, increased protein synthesis in early stage, cardiomyocyte hypertrophy, and increased ventricular volume and enhanced apoptosis. And the mechanism is still not clear. So current treatments include treating the condition's underlying cause, reducing symptoms, stopping the heart failure from getting worse, which increases lifespan and improves quality of life. And the treatments can be heart-healthy lifestyle changes. Guess that means no more steak. Ongoing care, medicines, medical procedures, probably no more steak and cheese. Medical procedures and surgery, there's an obvious need for new treatment options. And so there's a animal model for this, which uses isopretrenol. It induces maladaptive cardiac remodeling and heart failure in a mouse model. And so you get a lot of different changes, such as lipid peroxidation, oxidative stress, calcium overload, inflammation, and renin-angiotensin release. And so we set up these experiments, and Pepe actually is the one that designed these particular experiments, and she did it in such a way because she'd already done an experiment where she looked at protein turnover as a function of this layout. So what we're going to do is look at the protein synthesis and also protein turnover. So we have control, iso-treated, heavy AHA, light AHA. We're going to start labeling day 0 out to day 4 while we're treating them, and then we'll label again day 10 to day 14. And we have surgically implanted osmotic pumps to deliver the iso to mice over these 14 days. This is what the data looks like. So protein identification of day 4. These are replicates, so there's not great overlap among all the replicates. Day 14 time points, a little better of the replicates over this. So day 4, we're seeing about 1,500 or so proteins. being identified. By day 14, we're seeing a drop in the number of new proteins that are being synthesized and being expressed. So proteins with significant changes quantified day 4 and day 14. So the red bar is day 14, the blue bar is day 4. So we're seeing big changes in some places. There were 93 and 75 proteins that were significantly different between the ISO and control hearts in day 4 and day 14, respectively. So if you map these all out onto the maladaptive cardiac remodeling network, the red are identified in ISO only, the blue is identified in sham only. Sham is the control, and so you can see that's actually quite complicated. This is a comparison of acute day 4 and chronic ISO treatment day 14. Again, the red is day 14, the blue is day 4. And you can see there are many more pathways being challenged as a function of the longer time period for this. There are fewer in the day 4 treatment. This is a comparison of day 14 ISO treatment, day 14 control, day 4 ISO, day 4 control. We only get a correlation of about 0.69, but you wouldn't expect a very high correlation among these two data sets. You would expect increased number of changes as a function of the longer time treatment. So pathway analysis on the newly synthesized proteins with significant changes and unquantifiable large changes at acute and chronic time points, and correlation of NSPs quantified at both acute and chronic time points. So NSPs quantified at both acute and chronic analyses. So these are some of the proteins that are changing. This is one of the proteins up here where we see a very big change at day 4, but we are seeing some very unique changes at various positions in this treatment. And these are significantly altered, newly synthesized proteins previously associated with human cardiac diseases or animal models. So the ones that are in red are the proteins or genes that have been identified through human mutations as being involved in heart failure. The ones in black are human association studies, and then the ones in pink are from animal model studies. This is a combined analysis in day 14 with protein turnover data set. And so one of the things that Pepe had done and published back in 2014 is she did the same experiment, but she looked at protein turnover rates in response to ISO treatment. And so each trend is annotated with the top 2 significantly enriched pathways. And so we see so the ones that are in blue, we see an increase in newly synthesized proteins as well as an increase in turnover. The proteins that are in red, we see a decrease in protein synthesis, but an increase in turnover. The ones that are in green, the 11%, increase in synthesis, decrease in turnover, and then the ones in purple are both a decrease in synthesis and a decrease in turnover. So this provided a very interesting look at the data, because most of the time we're looking at just equilibrium of the protein expression. Here we're looking at both synthesis and turnover, and so we're looking at the 2 aspects of what results in protein equilibrium. So this is a comparison of the absolute NSP changes associated with each of the 4 NSP turnover trends. So most of the things that we see are involved in decrease in synthesis and increase in turnover. So just the highlights, we applied a new proteomic method to look at this heart failure model in animals. Proteomic data set revealed a complex combination of adaptive and maladaptive alterations at acute and prolonged time points, including the identification of proteins not previously associated with cardiac remodeling. The novel integration of the newly synthesized protein data set with previously published protein turnover rates in the ISO model demonstrated that alterations in specific biological pathways are produced by differential regulation of protein synthesis and degradation. This has just recently been published. My only foray into cardiology. Okay, so this is the early part of the talk. This was my group back in the old days. This is Jimmy Ng. While he might look like he just graduated from high school, he's actually has a master's degree in electrical engineering from the University of Washington. And I was looking for to hire a programmer, and on his CV, he had his electrical engineering master's degree used neural nets to try to develop I forget what it was. It was something to interpret speech or something of that nature. And when I was at Caltech, John Hopfield was there, and he had just reinvigorated the whole neural net field. And so I got interested in neural nets and tried to learn how to use them without much luck. And so his CV caught my attention, even though he was at the time working in a nursing home, running the computer programs for them. And that's Dave Schultz, Andy Link. Both these guys were heavily involved in developing the mud pit, as well as Ashley McCormick and Karen Johnshaw. This is a little later edition of Laura Hayes and Edwin Carmack. And that's a more recent picture of my group, although I'm not sure how many people are still there. We don't take pictures. I keep telling them we need to take a picture every year, and we never do. All right, thank you.
Okay, who would like to ask a question or two?
Hey, John. I just had a technical question on the bond cap method. How much more of the newly synthesized proteome do you see with that method versus just doing a pulse SILAC type of experiment and detecting the turnover?
So we haven't done that comparison. I'm trying to think of, so when you do just SILAC, you see everything, right? And so you're seeing some changes. It's when you do a very short pulse that you're really at a disadvantage with the SILAC strategy. There was a paper by a guy from the University of Manchester. He's now a dean. Simon Gaskell, where they did a short pulse, and they really struggled to see the signal from the noise with it, looking at things. We've done a comparison of using AHA with our light and our heavy AHA versus the strategy that Jerome Kriegsfeld came up with, where he used both a combination of SILAC and AHA. And we get, the results are fairly comparable. We do a little bit better, but. So I think the main issue is that you separate signal from noise much better with AHA. We've done much shorter pulses with AHA, looking at visual training and tadpoles with Holly Klein at Scripps.
While they're thinking about the next question, I might ask, so we've got 150 students of mass spectrometry here. What areas of the field do you think are fertile, and where would you, if you were a junior person, be looking?
So you didn't read my editorial in the last JPR?
Always. I want you to hear. I want them to hear from you.
So I pointed out that I frequently get asked that question, and the editorial that I wrote was in response to a workshop that I went to at Northeastern University last month. Actually, it was June. It's time's flying. It was a single-cell proteomics workshop, and very exciting. People have been dabbling with single-cell proteomics for quite some time. Now it's starting to get to the point where I think you can attack 1 million cells, things like that. And the people that were running the workshop are super enthusiastic about being as open as possible and sharing ideas, sharing technology, sharing methodologies, software tools, things like that, to try to get this field moving.
If I could follow that up, John.
If I could follow that up, so this area of conformational proteomics, in other words, instead of working with dead proteins like you've been doing for a long time, ignoring native mass spec, But other methods like, chemical labeling, cross-linking, applying those methods which previously were done with pure proteins to complex extracts, try to get an idea of what conformation of 20,000 proteins are rather than just one protein. Do you see that as possibly?
Are you reviewing my grants? So we just have a company in that editorial I just wrote is a perspective written by Nikolai Slavov on single-cell proteomics.
Can't believe you didn't read that.
It's out. It's online. And the next one that's coming is a perspective on proteomic level structural, or proteome level structural proteomics. And I guess the question would be, could we take HDX large scale? And I don't think so, because I think the computation's too complicated. So there are actually other strategies you can use to do sort of large scale structural proteomics. We're working on one right now, but there are some others. There's this proteolysis sensitivity that Paolo Piccotti's been doing. Sprox from Mike Fitzgerald. So there are other strategies that are out there for doing this. And I think that's kind of next level proteomics, because we're starting to understand how prevalent protein misfolding, how prevalent of a role protein misfolding plays in diseases, particularly as we get older.
100 years, Spuchner did it with a yeast cell extract, and that just 3 or 4 enzymes made a little alcohol. But you know what I mean?
Have you been drinking alcohol?
So the third perspective will be precision medicine. So that's another one that'll be interesting. I think that might interplay nicely with single-cell proteomics, if one could do that.
Any other questions from the audience?
And the fourth perspective will be
So I'm curious about your fascination with two-dimensional chromatography. There's a lot of guys and researchers out there like Gert Desmet and Pat Sandra, Dwight Stoll, who've really been pioneering some two-dimensional HPLC techniques, and I was just curious about the perspective of the role of 2D LC in proteomics, and also what the potential pitfalls are at this particular moment.
Well, I think I think the trick, and I'm not sure all the chromatographers get this, is that you have to make your chromatographic methods, you have to bend your chromatographic methods to the mass spectrometer, not the other way around. So you really want to deliver peptides in the best possible way for the mass spectrometer to do its job as best it can. And that the example that I gave is one of the illustrations of that. So the small scale chromatography, we were delivering low flow rate in the suitable buffer into the mass spectrometer. So the mass spectrometer could operate at its peak level. And I'm not sure that chromatographers always get that. So I'll tell you a little anecdote. Time to borrow.
Proceed.
So Arthur Mosley, he was a grad student in Jim Jorgensen's lab. So back in the 90s, late 90s, he was chasing me around. Talking to me to talk about this 2D LC stuff. He'd come up to me and he'd start asking me a question about how much carryover do we see, between steps and all this sort of stuff. And I said, well, not very much, maybe 10%, 20%. And I really wasn't getting at what he was asking. And so I finally had time to say, okay, what's your problem? And he says, I see massive amounts of carryover in my system from step to step. So how do you have your system set up? Well, he's got valves. He's got a desalting, offline desalting column, and he's running his reverse phase gradient around the ion exchange column rather than through the ion exchange column. And so what he hadn't realized is that the ion exchange media actually has dual characteristics. It's both an electrostatic phase and a reverse phase. And so unless you have a certain amount of organic in your buffer C, you still get retention of peptides by hydrophobicity. And we always ran our reverse phase gradient through the ion exchange column. And so we didn't see as much carryover. And so I pointed that out to him, and I never heard from him again. But as a trained chromatographer, he thought he knew better on how to do that. And he was not adapting his chromatography to his mass spectrometer. He was adapting his mass spectrometer to the chromatography. And I think that's the way you have to think about this in order to make it work.
Very interesting. One more.
Thank you for mentioning misfolded proteins and an implication in disease. I actually work with plants, and I work with ER stress in plants, endoplasmic reticulum stress. So the sort of gold standard for measuring ER stress has been transcript levels of UPR, unfolded protein response, and it's been really difficult to measure actual unfolded proteins or misfolded proteins in plants. Do you have any ideas, or is there something that I haven't read in terms of measuring ER stress directly using mass spec?
Well, we haven't published our paper yet. We're trying to get it published. What? So I could tell you how to do that.
I'm looking for PhD students.
Am I looking for PhD students? You know, I only ever have one PhD student, and he's gone. I call it, I know Josh has a ton of students. I call it stiff Lord training. So one master, one student. I have a lot of postdocs.
We're always looking for great students in Wisconsin, so. All right. Thank you, John.